My name is Jeff Johnson. I'm a professor of chemistry at Hope College, and my research focuses on the activation and functionalization of carbon-carbon single bonds. So if you look around you, anything that you see that is or once was living is composed primarily of organic material. It's going to be trees and plants, petroleum products, plastics, even your next door neighbor Fred, ultimately are all composed of organic molecules. And at the core of these organic molecules are carbon-carbon bonds. I like to think about those carbon-carbon bonds as the brick and mortar of organic molecules. So envision, if you will, a reinforced concrete wall. My mind immediately goes to an elementary school gymnasium. So you can paint those walls, you can decorate it, you can change the appearance, but if you'd really like to make a structural change to that wall, you might as well knock it down and start over. Okay, organic molecules are much the same. We can change various aspects of them and perhaps some reactivity, but if we want to change that basic core of that molecule, the carbon-carbon bonds, it's essentially impossible. If we want to make something like that, instead what we have to do is build it from the ground up. And so my group is hoping to change that. We're developing new methods using transition metals to break carbon-carbon single bonds through a process called activation, and then selectively modify those structures and react them with other species, called functionalisation. And so back to the wall, think about the prospect of taking out bricks and selectively putting in a door, or maybe easily taking out some other bricks and put in some windows. You can totally change the function of that wall, the original structure, without rebuilding it from scratch. We'd like to do the same thing with organic molecules. Nature provides complex organic molecules with a variety of carbon-carbon frameworks. We hope to use this new methodology for carbon-carbon bond activation and functionalization to modify these structures without completely rebuilding them. Ultimately, we'd like to make this methodology useful for groups across the country and around the world to use in their chemistry, potentially leading to new bioactivity, such as the next new antibiotic, or materials purposes, such as the next OLED for your next TV. The possibilities are endless.